This is the nucleus, and inside the nucleus we have the DNA. The DNA is made of 3 strands. In the strands we have the genetic information. The genetic information is like a code the DNA uses to command everything in our bodies. It's like the language that DNA communicates with the rest of the cell. The code is composed by 4 nucleotides. The disposition of these nucleotides will determine the commands of the DNA. Nucleotides appear on both strands complementarily. Adenine always pairs with thymine by 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds, each due to their unique structures. But the DNA can go out of the nucleus and speak with the cytosol, so this code must be transcribed and taken to the cytosol. This is called transcription. For this purpose, the hydrogen bonds are disrupted, and the sequences of the nucleotides will be copied by a complementary way. This will build the RNA from the DNA sequence. But the RNA doesn't have thymine to pair with adenine. Instead, adenine will pair with uracil. Guanine still pairs with cytosine. Now, RNA can take the DNA messenger to the cytosol in a code form. That's the reason this RNA is called messenger RNA. RNA goes out of the nucleus through the nuclear pore and enters the cytosol. Messenger RNA enters in the ribosome, so the ribosome can translate the code message from the DNA in a process called translation, with the help of its own RNA called ribosomal RNA. The messenger RNA fits in the ribosome and only 3 nucleotides are exposed. Each group of 3 nucleotides is called a codon. Each codon translates to only one thing, but the codon is not translated yet. For this purpose, another RNA will join the game. The RNA that transfers the translation of each specific codon. This transfer RNA will pair with messenger RNA inside the ribosome and leave one amino acid. Then all the information inside the DNA is encoded, and each codon is translated into one amino acid. The part of the transfer RNA that pairs with the codon from the messenger RNA is called anticodon. All codons will be translated into amino acids, which will combine to form a protein. Now let's come back to the nucleus. What would happen if the DNA got in contact with toxic substances like the ones present in cigarettes? They can damage the DNA and cause mutation, a change in the nucleotides. This mistake in the code will be transcribed, and the wrong messenger will be translated. The wrong amino acid will be in the protein, and the result could be a loss of function of this protein or a protein that works in a bad way.